The first step to Illumina DNA sequencing is sample preparation. This is necessary in order for the following steps to occur. There are a few ways to prepare samples. The process that is used in Illumina DNA sequencing begins by first starting with extracted DNA. Next, transposons fragment the DNA and adapters are added to the ends of the strands. This process is called tagmentation. Then, the adapters are ligated, meaning they are put together. Lastly, through reduced cycle amplification, sequencing binding sites and regions complementary to the flow cell oligo are added. Oligonucleotides are short nucleic acid polymers which are used in research. The next step that I'm going to talk about is the first read of DNA sequencing. Sequencing begins with an extension of the first sequencing primer to start the first read. Nucleotides that are each marked with a certain color light then start to look for a spot on the chain to connect to. However, only the correct complementary base will connect to the strand. The certain lights are given off when the nucleotide attaches to the sequence. This is called sequencing by synthesis. This keeps happening for a specific amount of time, which creates a read. The color and intensity of the light that the nucleotide emits identifies the complementary base. These strands can be read and clusters are sequenced in a parallel process. Lastly, the product from the first read is removed.